Tissue culture. In plant breeding, the technique of tissue culture has emerged as a very efficient tool to propagate better improved crops. Tissue culture is a much approachable technique than other traditional plant breeding techniques. By using this method, cells, tissues, etc. are maintained and grown in vitro under aseptic conditions in artificial medium, providing necessary nutrients for growth. In 1839, Schladen and Schwann proposed that cell is the basic unit of life and is capable of autonomy. Each cell, if given an environment, is capable to regenerate into a whole plant. Based on this theory, a German physiologist, Gottlieb Heberland, in 1902 developed the concept of in vitro cell culture, also called tissue culture. Cells in his experiment increased in size but failed to divide, due to which his predictions failed. However, his hypothesis paved the way for more research. He is thus regarded as the father of tissue culture. Taking cue from his experiments, Hanning in 1907 took nearly mature embryos of crucifers and successfully grew them to maturity. In 1908, Simon regenerated callus, buds, and roots from poplar stem segments and established the basis for callus culture. A breakthrough came from White in 1934, who experimented on tomato roots. He reported not only could culture tomato root tips grow, but they could be repeatedly subcultured to fresh medium of inorganic salts supplemented with yeast extract. During 1950s, scientists found that any part of a plant grown in nutrient media under sterile conditions can give rise to a whole new plant. The plant part used in culture is called as explant. This property of plant cells to give rise to a whole new plant is called totipotency. The term totipotency was coined by Stewart in 1968. Totipotency. It requires a nutrient medium that contains sucrose, inorganic salts, vitamins, proteins, and growth regulators, such as auxins and cytokinins. Technique of tissue culture. The first step of tissue culture is excising or cutting a small part of plant tissue, like the leaf, root, or shoot of a plant. This cut-out part, explant, is then placed in a suitable nutrient medium containing a carbon source such as sucrose, inorganic salts, vitamins, amino acids, and growth regulators like auxins, cytokinins, etc. The explants are allowed to multiply and form a group of tissues called callus. Callus is irregular, unorganized, and undifferentiated mass of actively dividing cells. This callus is then allowed to multiply to form many plantlets, which are then transferred to a different container for rapid multiplication. Finally, the developed plantlets are transplanted to the soil for normal growth. Tissue culture is performed using a small part of a plant to develop a large number of plants in a short duration. It is thus also called as micropropagation. As all these plants are developed from the same part of the parent plant, they are genetically identical and called soma clones. Micropropagation is extensively used in horticulture and floriculture and is employed in the large-scale production of tomato, banana, apple, etc. This technique can also be used to propagate high-yielding disease-resistant varieties in a short span. Even in conditions when a plant is diseased, tissue culture is used for recovering healthy plants from diseased plants by taking the explant of the apical or axillary meristem. This is done because the meristem is always free from pathogens due to lack of vascular supply. The process of using the meristem for propagation is known as meristem culture. Culturing of meristems has been successfully carried out in plants such as banana, sugarcane, and potato. There are many advantages of tissue culture, which are as follows. A large number of plants can be grown in a short period of time. Healthy plants can be grown from diseased plants. Plants without seeds can be multiplied. It is useful where sexual reproduction is not possible.